Life. Evolution. Is there a hidden order to things, or is it all chance? Randomness. What does it mean for the world to be a matter of chance? These are big questions, shadowy questions, elusive questions. You think you know what survival of the fittest means? Well, we'll just see about that.
The origin of life.
Let's take a look at how life most likely arose here on Earth.
It is suggested that 3.5 to 4 billion years ago, the atmosphere on Earth was made up of methane, ammonia, hydrogen, and water vapor. Basically, a lot of gas, not a lot of life. In fact, no life. But then
these things we call protocells formed. The first life on Earth. Proto just means first, or earliest form of. So protocells were the earliest form of cells.
There are a lot of different ideas people have about how exactly these protocells came about. For example, some people think they came from outer space. Woo! Anyhow, regardless of where exactly they came from, the basic gist is that some amino acids that were floating around got heated up by the gases and then bonded into polypeptides, or proteins. These were the first protocells, and that's how life got started. Pretty random, huh?
You can just imagine billions of protocells floating around. Then, one day, about 3.1 billion years ago, one of those protocells developed a helpful protein, and this special cell began to reproduce. In fact, that cell is the common ancestor of all living things on Earth. That's right. Every plant, animal, person, and microorganism started with that little protocell. We all have the same origins.
Now, those first cells were prokaryotic cells, a primitive type of cell that lacks a defined nucleus and most organelles.
But what about eukaryotic cells? The more complex kind that you, me, every plant you see, and mushrooms are made of. How did those come about?
Actually, we're not 100% sure about that. But there is, of course, a theory. It's called endosymbiosis. It's a scary word that describes a creepy occurrence. According to endosymbiosis, some of those prokaryotic cells swallowed some other prokaryotic cells, and those swallowed cells developed into the organelles of the cells that had swallowed them.
Oh my gosh, that is so creepy and gross. It's like if my friend Emma swallowed me and then turned me into her stomach so I could digest all her food for her.
Yeah, more or less. But remember, before said swallowing, those simple prokaryotic cells didn't have many, if any, organelles, so they weren't able to do complex things like digestion. By developing organelles through endosymbiosis, they became the complex cells known as eukaryotes. Of course, this happened over a long period of time. Creepy, but kind of cool.
We've got prokaryotes and eukaryotes, right? The prokaryotes are the simple guys with no nucleus or organelles. And the eukaryotes are the more complex cells. Somewhere along the line, scientists realized that among the prokaryotes, there were these two types that were just too different from each other to belong to the same group. So they divided them into two groups, bacteria and archaea. So if we wanted to construct a tree of life showing these different life forms, it would be something like this. There's three separate categories: eukaryotes, bacteria, and archaea.
Although the archaea, like bacteria, lack organelles and a nucleus, it was discovered that the ribosomal RNA is actually more similar to the eukaryotes. Archaea don't require light or oxygen and can therefore live in the most extreme of environments. They may be the oldest life forms on Earth.
And that's the origin of life on Earth. Remember next time you go to step on a spider or swat a fly. Those guys are made from the same stuff as you are.
Evolution. Setting the stage.
Let's take a look at one of the most important concepts in modern biology, evolution.
Madame Vocabulary here. Your future is evolving, traveler. Prepare for change in the next 40,000 years. Here's some vocabulary to get used to in the meantime.
Evolution is a change in the number and types of alleles present in a species over time. An allele is a version of a gene, like brown or blue for eye color. Heredity is the passing down of traits from parents to offspring from one generation to the next. Mutation is when a change in a gene's DNA occurs. Variation is differences in appearance in members of the same species. Natural selection is when organisms with more successful phenotypes for a given environment survive to pass their alleles down to their offspring.
First, let's define evolution.
It can be a slippery topic, but the simplest and yet most widely accepted definition is that evolution is a change over time in the types of alleles present in organism populations.
An allele, remember, is a form of a gene, like brown versus blue eyes for eye color, long stem versus short stem for pea plant stem length, or fire-breathing versus non-fire-breathing for dragon breath. There's always one of the two versions a gene can take, and those versions are called alleles. Got it?
Okay, so evolution, once more, can be defined as changes in those alleles over time. That's all it is. As these changes accrue over long periods of time, they cause great changes in organism populations.
Let's talk about a few basic ideas that are crucial to evolution. First up, heredity. A parent organism passes traits down to its offspring. In other words, the offspring inherits these traits. Easy.
Next up, variation. Within a population, individuals have different traits. They're not all the same. That would be no fun. Some of us humans have brown hair, some have blonde, some have red or black. That's variation. All plant and animal species have variation.
So, since traits vary and since offspring inherit traits from their parents, it follows that the variation among traits gets passed down through generations. This is simple, obvious stuff, but these things are the fuel that evolution runs on.
Okay, now let's dive into the poster child of evolution, natural selection. This is an important idea that people sometimes confuse. It's actually really simple as long as we have our facts straight. So let's get them straight, once and for all.
Natural selection, a term first introduced by the famous naturalist Charles Darwin, says that those individuals with beneficial traits, such as good eyesight or fast reflexes that increase hunting ability, will survive, produce more offspring, and thus contribute a larger percentage of their genes to the next generation. Those with unbeneficial traits, such as being slow or weak, do not survive or reproduce. So the beneficial traits are the ones that end up getting passed down and the ones that end up being prominent among a species.
Let's say we've got some elephants that live in a land where it rains 90% of the time. Some of the elephants have waterproof skin, and some don't. The ones that don't have waterproof skin get sick from the rain and die out. The gene for waterproof skin ends up getting passed down, while the gene for non-waterproof skin gets phased out. Sounds practical to me.
Another way you might hear natural selection described is as survival of the fittest. This phrase can be a little misleading unless it's understood properly. Let's talk for a little moment about what is meant by fittest.
Fitness describes how much an allele affects an organism's ability to survive and reproduce in its environment.
Exactly. In its environment. The fittest does not necessarily mean the strongest or the biggest. It just means whatever works best given the situation. Survival of the fittest could really go survival of the most well-suited to a particular environment. The waterproof elephants were considered the fittest because they happened to live in a place where it rained all the time.
Say the elephants lived in the desert, and the waterproof skin was so heavy that those who had it died of exhaustion. In that situation, the elephants with waterproof skin are not the fittest, and the genes for that trait get phased out.
As a child, I tried to explain to my mother I was not well suited to live in a mountainous region. I was just too clumsy and darn near tottered off a ledge on several occasions. I suppose she thought I would grow up to be an acrobat.
Before we move on, let's mention two more very basic things to keep in mind about evolution. First of all, remember that evolution happens at the level of the population. That is, individuals do not evolve. Only populations evolve. A population evolves over many individuals' lifetimes.
And finally, my favorite part of evolution is that evolution is random. Individuals do not learn new traits to adapt to their environments, and there is no ultimate plan for what a species will look like in the next million years. Kind of like a plastic bag being carried by the wind, species react to whatever factors come their way, and these factors are random. We'll learn more about the factors that affect evolution in the next section, so keep in mind the randomness of it all.
Evolution in action.
Here is some vocabulary to help you in your future, travelers. I require one pinky toenail and a handful of marbles for my services.
Genetic drift is a change in the frequency of an allele's occurrence in a population due to random chance. Gene flow is when two isolated populations swap genetic information. Speciation is the formation of a new species.
We're going to check out 3 ways that evolution can occur. That's natural selection, genetic drift, and gene flow. First up, natural selection. Let's go for it.
Attention! Natural selection is the most common way that evolution occurs. Let's talk about it. We already know that natural selection is the process by which organisms with traits that are beneficial in a given environment survive to pass those traits down to their offspring. Over time, these traits become widespread features of the species.
Here's a great example that makes it really easy to understand natural selection and evolution. London, England has this species of moth called the peppered moth. Back in the day, most peppered moths were white. This helped them camouflage against light-colored trees and lichens so their predators couldn't see them. But then, the Industrial Revolution happened and all of London, right down to the leaves on the trees, became covered in this gross black soot from all the factories. So, what happened? Well, the few black peppered moths in the population became the ones that got camouflaged, and they started to thrive while the white ones got eaten.
This just in, fashionistas. Black is in, white is out. What will happen tomorrow? Stay tuned!
Another thing that is a factor in natural selection are resources. Resources are limited in nature. There's not always enough food to go around for everyone. So, some individuals within a population will not get enough resources to survive and/or reproduce. Therefore, individuals differ in how many offspring they produce for the next generation.
Natural selection changes allele frequencies over time, leading to distinct shifts in the phenotype and therefore the genotype of the species. In other words, natural selection brings about evolution.
Notice that natural selection is the result of a condition in the environment, like an ice age or global warming. This is important to remember as we discuss some other ways evolution can occur that are quite different.
Also notice how natural selection acts on the phenotype rather than the genotype of an individual. Remember, the phenotype refers to the traits being expressed, or the ones you can see, while the genotype refers to the genetic makeup. So naturally, selection has to do with the organism's interaction with its outward environment. It makes sense that change occurs via the physical features it expresses, or the phenotype. In our example of the peppered moth, the black color of the moth is the phenotype that thrived in the environment of soot.
Another way that evolution can occur is by genetic drift. Let's go there.
As opposed to natural selection, which is when evolution occurs due to a condition in the environment, genetic drift is when evolution occurs due to random chance.
The effect of genetic drift is small in large populations, but large in small populations. So we'll use a small population to start. Oops! These 4 rats fall off a cliff. These 4 survive to reproduce. The genes of the population from here on out are only going to come from these 4 rats, so whatever traits they've got, the future generations will get. Any traits that only the ones that fell had are no longer in the species gene pool. That's genetic drift.
Genetic drift can create a change in allele frequencies over time. As we mentioned, this effect is much more pronounced in small populations than in larger populations. Remember, genetic drift is due to random chance, while natural selection is driven by the fitness of certain alleles in a given environment.
Here are 2 extreme examples of genetic drift: the population bottleneck and Fanders effect.
A population bottleneck occurs when a very large percentage of a population is wiped out, leaving the rest of the species to develop from the genes of the small group of survivors.
Did you know that there is a great example of this bottleneck effect in human history? Based on studies of mitochondrial DNA, Y chromosome DNA, and lots of other genes, it is estimated that our latest population bottleneck occurred about 70,000 years ago, around the time that the Toba supervolcano exploded in Indonesia and wrought massive destruction. Everyone in the world today is descended from a population of 15,000 people who survived. Consequently, and due to several other bottleneck episodes, humans have much less genetic variability as a population than do all other primates.
The other extreme example of genetic drift is Founder's effect. Founder's effect occurs when a small subgroup splits off from a larger population, and the smaller population ends up with a different genetic makeup than the larger group. In this case, unusual genes can become the norm, since they represent a higher percentage of the gene pool. Even if one person has an unusual allele, it can become highly represented in this smaller group.
A well-studied case of Founder's effect occurred in the Pennsylvania Amish, a group who broke off from the original European population. Two of the original founders carried an allele for Ellis-Van Creveld syndrome, which is characterized by heart defect, skeletal defects, and polydactyly, which means extra digits on the hand or foot. The prevalence of this allele among the Amish is many hundreds of times greater than any general population. So interesting.
So genetic drift is based on random chance. Think about it like this. A species is traveling down its evolutionary path, la-di-da, following the twists and turns of natural selection. Say a new path suddenly sprouts off the main one in a different direction. This new direction represents a drastic change in the possibilities of the gene pool. Notice that, as opposed to natural selection, genetic drift occurs in the gene pool via a change in genotype rather than phenotype.
Evolution can also be affected by something called gene flow. That's our next stop.
Gene flow occurs when individuals move between isolated populations and swap genetic information, thus increasing the amount of genetic diversity as opposed to genetic drift, which decreases the amount of genetic diversity.
Gene flow was rare in prehistoric times. Think about it. Cavemen didn't have cars or planes to get around, so they dated locally. Genes from different tribes didn't often get mixed. Instead, tribes evolved in isolation.
Say we've got a tribe of triangles here. They live down in a lonely little valley. And here's the circle tribe. They live on the other side of this huge mountain chain from the triangle tribe. The mountains prevent the two tribes from ever interacting. Well, one day, a brave triangle dude ventures over the mountains, ends up falling in love with a circle woman, and they reproduce. The child now has circle and triangle genes. As the child grows up and procreates, further genetic mixing will occur and move down through the generations.
So gene flow is another way that a species can evolve via genotype. It's like a genetic bridge being built between two isolated groups. The bridge makes it possible for the two groups to swap genetic info. This new information then gets recombined into each tribe, diversifying its gene pool.
We've just talked about natural selection, genetic drift, and gene flow, three things that fuel evolution. Eventually, natural selection and genetic drift can lead to the emergence of a new species. This process, called speciation, is one of the most exciting events in evolution. How does it happen? Well, either one species splits into two new ones, or one species becomes radically different from what it was before.
Gene flow, as opposed to natural selection and genetic drift, counteracts speciation. Think about it. If you have two species developing in isolation, they are distinguishing themselves more and more from each other. Maybe the circles are getting rounder and the triangles pointier, but when they mix, their genes are blending and the distinctions between the species are being blurred. That's anti-speciation.
Now that we've learned about these ways that evolution can occur, let's dive a bit deeper into some evolutionary action.
Okay, first, let's talk a bit more about variation. Remember, all plant and animal species have variation among individuals, and this variation is passed on to offspring and down through generations. As we know, variation is increased by recombination of genes on chromosomes during sexual reproduction. This occurs to the chromosomes during crossing over, the part of meiosis when sperm or ova are made. Genes are recombined with other genes, leading to interactions between the genes, the results of which may reinforce or suppress the activity of a gene or protein. You're probably thinking, so what?
Well, in a constantly changing world, variation can prove very beneficial to species, increasing the likelihood that it will survive under new environmental conditions. Think about our peppered moth. If the moths were all white and didn't have any variation, the entire species would probably have died out during the Industrial Evolution. But since there was some black ones, these were able to thrive under the new conditions and preserve the species.
Another factor to consider in evolution is mutation. Mutations are constantly popping up in the gene pool and increasing variability in a species. Although most mutations are not beneficial, some are. Let's take a look at an interesting example of how a mutation affects natural selection. Pay attention, this is really cool.
But first, travelers, let's peel back the layers of time and reach into your deepest memories to review a few terms. Close your eyes, close your eyes.
A homozygote is an organism that has two identical alleles for a particular gene, like two dominant alleles or two recessive. A heterozygote is an organism that has two different alleles for a particular gene, that is, one dominant and one recessive.
Wake up!
Okay, so let's check out this mutation. Here we go. It's a mutation for sickle cell anemia, which is a recessive trait, and it's found in people of sub-Saharan African descent. This mutation is mega unbeneficial to the homozygous recessive folks. This is where it gets interesting, so listen up. Because most recessive homozygotes used to die before they reach reproductive age, this trait should have become highly selected against, right? The sickle cell allele should have become extinct in the population, or at least exceedingly rare.
But that's not what happened. Instead, about a third of all people with ancestors in the sub-Saharan African region are heterozygous for this allele. That's very peculiar. Whenever something is peculiar, scientists take notice and investigate. Let's follow the investigation.
Sickle cell trait is the result of a change in one amino acid, resulting in a mutation in the hemoglobin in red blood cells. These sickle-shaped blood cells can obstruct capillaries, leading to blood loss in organs and damage to these organs. In the heart, it causes a heart attack. In the brain, clots in the blood vessels cause strokes.
So, how did this very unbeneficial allele survive? Why would it be so widely distributed and highly represented?
Well, it turns out that the heterozygous genotype is highly selected for. But why? Because people who have at least one allele for the sickle cell trait are much more resistant to malaria than people who have no sickle cell alleles.
So people who have no sickle cell alleles died of malaria, while those who had two sickle cell alleles died of sickle cell. But the heterozygotes, who had one of each, survived malaria and showed no phenotype for the sickle cell disease. Since it's the heterozygotes carrying one sickle cell allele that lived, the allele continued to get passed down.
That's pretty amazing. The sickle cell allele is so effective at increasing the survival in malarial areas that disease-causing allele has mutated and arisen at least 5 times in human history.
And that's evolution in action for you, folks.
The Hardy-Weinberg equilibrium.
What is the Hardy-Weinberg equilibrium? Some super cool band nobody has ever heard of? No, it's the concept that genotype and phenotype frequencies stay the same in a given population unless they are acted upon by an outside force. Well, no offense, but duh. If nothing happens to change the allele frequencies, they won't change. It's kind of obvious.
Hey, wait. What are genotype and phenotype frequencies?
Frequency just means the rate at which something occurs. So allele frequencies are the rates at which alleles occur.
Let's see what kinds of things can happen to disturb the Hardy-Weinberg equilibrium. We've got non-random mating, mutations, natural or artificial selection, limited population size, overlapping generations, random genetic drift, and gene flow. Basically, anything that can change gene frequencies will change gene frequencies.
So in reality, the Hardy-Weinberg equilibrium, which assumes that allele frequencies don't change over time, is impossible in nature. Think about it. If allele frequencies don't change, no evolution would occur. And where would we be then? We'd probably all die out.
So, if it can never really happen, why even bother with this Hardy-Weinberg equilibrium? Seems kind of unnecessary. Just saying. Right?
Well, you see, the equilibrium represents an ideal. Even if it never actually occurs, it is helpful for scientists to talk about and compare things to. Since evolution and change are constantly occurring, the Hardy-Weinberg equilibrium is like the still point in a turning world. It's the only way any measurements can be taken. Let's take a look at an example.
Okay, the simplest example is a single gene with two alleles, one dominant and one recessive. Let's see, what kind of animal could we use? How about vampires?
Why is everyone so interested in vampires these days? Why, in my days, we kept our vampires locked up in the cellar and only brought them out on holidays.
All right, let's look at our example with vampires. Blood-sucking is dominant to non-blood-sucking vegetarian vampires.
The frequencies for the prevalence of big B and little b are P and Q, respectively. You always use P and Q for gene frequencies in Hardy-Weinberg equilibriums. Why? That's just the way it is.
Here's the first equation. It's kind of an easy one. The frequency of the dominant allele plus the frequency of the recessive allele equals 1. That's because you can think of the frequencies as percentages. And the percentage of the big B in the population plus the percentage of the little b in the population equals 100% because there are no other alleles.
Here are some other very useful equations to know. P squared is the frequency of dominant homozygotes in the population, while Q squared is the frequency of recessive homozygotes in the population. 2pq is the frequency of heterozygotes in the population. So how does this help us?
We only need to know one number to figure out all the rest of them. The number that you're usually given is the number or percentage of recessive homozygotes in the population, because that's the number you can see and count most easily.
So, getting back to our vampires, in a population of 1,500 vampires, 1,488 of them are blood-sucking. How many are heterozygous for the blood-sucking trait? Let's use our Hardy-Weinberg equation to figure it out.
First, let's note that if 1,488 of the 1,500 vampires are blood-sucking, then 12 are not. Now let's go from there. Pay attention, a little math going on here.
In this case, in our population of vampires, 12 out of 1,500 vampires are vegetarians. Vegetarianism is the recessive trait. So we know for sure that the vegetarians are the recessive homozygotes. Convert that to a percentage by dividing those two numbers, and you get about 0.008, or 8%. That's equal to Q squared.
Now a teeny bit of algebra. To get Q, which is the frequency of the recessive allele, just take the square root of both sides. There it is.
Here's that easy equation again. Solve for P, and you find the frequency of the dominant allele is 0.911, or 91.1% of the alleles in the population are for blood-sucking. Would you have expected anything less?
From there, you can calculate how many dominant homozygotes and heterozygotes are in the population. For dominant homozygotes, find P squared. The frequency of the dominant homozygote is 0.830, or 83%. Considering that there are 1,500 vampires in the population, it follows that 1,245 of the vampires are homozygous dominant, since 83% of 1,500 is 1,245.
To calculate how many heterozygotes are in the population, find 2pq. The frequency of the heterozygotes is 0.162, or 16.2%. Considering that there are 1,500 vampires in the population, then 243 of the blood-sucking vampires are heterozygotes.
An easy check for your work is to add up all the vampires and make sure it equals the original population. Add the numbers of dominant homozygotes plus the heterozygotes plus the recessive homozygotes, and you should get a number plus or minus 2 of the original population. 1,245 dominant homozygotes plus 243 heterozygotes plus 12 vegetarians equals 1,500.
And that's what you can do with Hardy Weinberg.
Now, my friends, nothing to worry about. We all know that vampires don't actually exist. Oh. Where'd the lights go? I'm scared.